What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going to dive into a review of molecular genetics, talking about DNA, RNA, protein synthesis, the central dogma, all kinds of fun stuff. So let's get right into it. So remember, DNA, kind of for its background, what it does is it contains genes, and on those genes is information on how to make proteins. So all about DNA is all about information, not only about us, but the proteins that make us. And where can we find DNA inside of the eukaryotic cell is always inside the nucleus. In prokaryotic cells, it'll be out free floating in the cytoplasm. That's kind of a difference between prokaryotes and eukaryotic cells, is the location of the DNA. And in eukaryotic cells, that'll be located in the nucleus. Now, DNA does have an overall structure that kind of looks like a spiral ladder, and this is called a double helix, where it has two strands that are kind of bound together, and they're complementary strands, where one is kind of the opposite of the other. What makes a strand is these things called nucleotides. So all these little colors that you see right there, those would represent one of the bases of a nucleotide, and the nucleotide will have a ribose sugar on it as well as a phosphate group to kind of connect to the next one and make these chains down their two strands that they have. So what those strands look like, here we go, you can see a nice nucleotide here with this adenine group with the phosphate, the 5-carbon sugar, which would be deoxyribose for DNA, and then the nitrogenous base here of adenine bound into the thymine. So adenines always pair up with thymines because they make a nice little match there. You got 2 hydrogen bonds. You have guanines pairing with cytosines, so the blue pairs with the red here. They got 3 hydrogen bonds there, so they are a nice grouping together. So it's all based on their hydrogen bonding and their base pairing. So A's always go with T's within DNA, and G's always go with C's. All right. Now, replication overall is the copying of DNA. And this is done during that S phase of cell division. So when the cell's going through its interphase, it's making that S phase, where it's making that duplicate copy of its DNA, that's the replication process. And it's known as semi-conservative because you're actually taking one old strand, you're going to keep it and use it as a template to make a new strand off it. So we take the original strand of DNA that's here, we unwind or open it up, so it exposes the 2 template strands now, and then we build new nucleotides off of that to make a complementary base pair. So it's a one old and a one new kind of scenario with base pairing. All right, so how does this process overall work for DNA replication? Well, it has 4 steps that are involved in it. The first step is to open up that helix. If you notice that we had to unwind it to get it to open up, and I'll show you a picture again in a moment. And the enzyme that does that is called helicase. So helicase will unwind or open up that double helix strand, expose both of those strands to be templates to allow some other things to make the base pairing off of it. Then a small section of RNA will actually be put in there, and RNA will kind of serve as a placeholder, in this case as a primer, to let the different enzymes know, hey, this is where we start copying the DNA, and that's called primase, the enzyme that actually puts that RNA group on there to get it started. Then what will happen is DNA polymerase will come in and basically start filling in and base pairing accordingly. Now, DNA polymerase will always work in a 5 to 3 direction. So if you ever see on DNA it labeled in 5 on one end and 3 in another, that's telling you which direction that the polymerase would work. So DNA is always replicated moving from the 5 end towards the 3 end, okay? So DNA will base pair up, A's match with T's, G's match with C's, etc. And sometimes it makes mistakes, or there's little gaps that need to be filled, and that's where another enzyme comes in behind it called ligase, and it'll check for errors, make some small edits, or whatever needs to be done to get the whole thing to work. So it's helicase to primase to polymerase to ligase, and it's always done in a 5 to 3 direction. All right, so again, seeing this whole process holistically, we can see the chromosome, this condensed version of DNA, but we'll unwind it as chromatin here, getting it nice and relaxed. And then this helicase enzyme will come in and break up the double helix, exposing out the 2 template strands. DNA polymerase will lock on and start building the new strand off of it. You can see the nucleotides being added in here. And then this one's working backwards, and that's because this is called a lagging strand. Because it's 5', it always moves from a 5 to 3 direction. It's 5 to 3 going this way, but also on the bottom it's 5 to 3 going this way. So actually, this polymerase has to constantly work in chunks. That's why it gets this name of a lagging strand, where it's constantly lagging behind helicase that's opening up the helix. This one is called a continuous strand because it just continually follows helicase all the way through replication. All right, so that brings us to a different type of nucleic acid called RNA or ribonucleic acid. This one has a little bit of subtle differences from DNA. So I have DNA here on this side and RNA on this side. You can see DNA is a double-stranded molecule, while RNA is a single-stranded molecule. Also by their name, we have ribonucleic acid here, and this one is called deoxyribonucleic acid. So there is a difference in the 5-carbon sugars that they have. There are some differences in their bases. Instead of having A's, T's, G's, and C's, RNA has A, U's, G's, and C's. So we actually lose the thymine that does not exist in RNA, and we replace it with something called uracil. So in here, the base pairing will function where A's base pair with U's and U's base pair with A's. They don't have T's like the DNA does. So let's kind of look at some base pairing overall. So if DNA is a T, an A, and a C, that means a complementary RNA would have to be a U, and a G, because here T's can exist in DNA but not in RNA. So it's going to base pair with an A with an RNA, because A is a valid letter for RNA, but this A that's in DNA has to base pair now with a U. G's and C's always stay the same. All right, so if RNA says U, G, and A, the complementary DNA would be A, C, and T. And then if the DNA, just to kind of throw you off here, so if DNA is T, A, and C, that means the corresponding DNA that goes with it would be A, T, and G. So just a little practice with base pairing. Remember, A's go with T in DNA, A's go with U's in RNA, and G always goes with C. All right, so this brings us to this idea called the central dogma. And this is basically the process of making protein. Sometimes it's just called protein synthesis. And the reason it's called the central dogma is it is the foundational principle for all things in biology, where you must go from DNA to be transcribed to RNA to be translated into protein. So the information that's held within the nucleus here as DNA needs to have a buddy kind of help move that information out, and that's the role of RNA. And then protein is a different overall macromolecule. There's got to be a translation, kind of changing these languages here. All right, so there are some different types of RNA that kind of accomplish this job. The first one is the messenger. The messenger is basically the one that reads the DNA in the nucleus and takes that message in a single-stranded version out of the nucleus. So it can be read a little bit later and actually build a protein from it. So the way that it's built is the DNA is opened up, there's complementary base pairing that functions there, and then that RNA strand is able to leave the nucleus, carry the code with it, and go find what's called a ribosome to be able to build a protein. Now, at the ribosome, another type of RNA is brought in called tRNA, and tRNA's job is to transfer over an appropriate amino acid to build the protein. So it actually takes the code that is in nucleic acid in RNA, and it matches it up with the corresponding amino acid, which is the language of DNA. So it's kind of like a little bit overall of a Rosetta Stone, where it's matching two different languages and bringing the appropriate thing over to build the new thing. All right. So basically how it kind of functions in here is the tRNA obviously has these nice little series of nucleotides on it, and it'll match up accordingly to base pairing to the mRNA. So if I had CCC here, it'll match up with GGG, and it carries with it here one of these amino acids. So you can see them being brought into the ribosome here. So this new one that's coming in is going to base pair up, and then it's going to bring over its amino acid to be added to this chain, and that will eventually fold into our protein overall. So this is all being done at the ribosomal level, this protein being built, but it's all based on the code that's brought over by mRNA from the nucleus. Okay, another thing that we can call mRNA is called a codon. So a codon is just another name of mRNA, specifically a 3-nucleotide sequence that gives us an amino acid. So every 3 nucleotides equals 1 amino acid. So when you have an mRNA sequence, you can kind of read it in groupings of 3, and each grouping of 3 will equate to an amino acid from our codon chart. So the codon chart is always used with the mRNA, because that's what we're using from the DNA. That's the message that's coming from the DNA, so that's ultimately what is used to build the protein. So that's what we will use in these charts. Now, the charts, the way we read them is we start from the inside and we work our way out. So if I have a sequence of UUU, right, as my mRNA or my codon sequence, I'm going to look at the chart from the middle here. I'm going to go to U, this first one, the big one. Then I'm going to go to the next U here, and then the following U underneath it to get phenylalanine. So you start from the middle and you work your way out. So U was the first example I had there, but we could do A, G, and C. So you start at the A, you work your way to the G, and then finally the C to get serine over here. And then ultimately another example is U, G, and A, and that'll give us a code that says stop. So basically when you reach that mark there, it breaks off, the ribosome releases the mRNA, no new amino acid is added, and the protein is kind of finished up, it folds on its own, and it does whatever it needs to do in the cell. There is one that's interesting, it's A, U, and G. Methionine right here, that is the codon that's at the beginning of every part of protein synthesis. So technically every protein gets the same amino acid at the beginning, and that's methionine. The protein can, this amino acid can be within the protein again at different points, but it always starts with methionine. And it always ends at a UGA, a UAA, or a UAG. Those are the different stop sequences. All right, so what can changes in DNA lead to? So now we talked about protein synthesis. What would happen if we had some changes in DNA? We get what's called a mutation. Now, not all mutations are bad. Really, all they are is just any change in DNA. So this could lead to a change in an amino acid that's being put in a protein. If you think about how proteins are being built, they're being built because the mRNA is being read, the mRNA is being read at the ribosome when that mRNA was produced by base pairing in the DNA in the nucleus. So if the DNA changes, that changes the message that's coming out, the code that is coming out, and then the amino acids could change that are being built in at the ribosome. Excuse me. All right, so there could be a lot of changes too. So we could have maybe one change, we could have actually no change as a possibility, and we could have a bunch of amino acids change that might actually build a new protein that functions better, could build a protein that functions worse, or a protein that doesn't function at all, or literally made no change at all in the protein. So those are all different outcomes. So more than likely, what's gonna happen with a change that happens to DNA is nothing, and we'll talk about why in a little bit. But some different things that could happen are called substitutions, where maybe their DNA sequence has a nucleotide that's switched out. As DNA is being copied, there's a chance for errors. If there is an error that occurs, it could produce a different outcome. So let's say we have the sequence of AAT, that's supposed to give us a leucine. Instead of AAT, now it switches to AGT, and instead we end up with a serine. That could change the function of the protein, or it could make the protein better, it could make it worse, or it could really do nothing for the protein. It all depends on the location of this change and what it does to the protein ultimately. But this is a change in the DNA, so technically a mutation. Another change in DNA is where you can insert something. So if you put in an extra amino acid, an extra nucleotide in there, it can have some implications on the amino acids. Now, sometimes you get what's called a frame shift, and I'm going to focus on that one real quick. So if I add a nucleotide in here that wasn't here before, so you can see it's kind of put in red, right? Now everything that's coming after it is read differently. If you notice, everything got bumped to the side. So this is where the shift in the frame occurs, right? It's not reading it in the same groupings of 3 as it did before, now it's shifting it over. So as a result, instead of having the glycine, proline, threonine like it's supposed to be, now it's glycine, serine, histidine. That might actually lead to some long implications for the protein. But ultimately, you do know that it did change, right? Proline is not serine, and threonine is not histidine. So those are ultimately different. Another thing that could happen is you could pop a whole extra codon in there, and this can happen. And as a result, if one whole codon gets put in, then it's just going to change that one amino acid. Basically, just insert an extra amino acid in there and space it out. So this could be a smaller change, but this one is a frame shift. This will change basically everything after it gets messed up. Another one is deletion, where you can actually remove pieces of the DNA. So this is actually showing it as a large-scale deletion, maybe at a chromosomal level, where a large chunk of DNA gets erased for some reason. It could happen where a single nucleotide gets erased. Just like before, with insertion, you can have those frame shifts that occur with deletion, and it ultimately leads to long changes within the amino acids that come after it. Translocation is a tricky one. This is usually where whole sections of DNA get flip-flopped. It's kind of a never a good situation typically, but you can have these like chunks of DNA from these different chromosomes here for some reason get flipped. Let's say this is chromosome 11 and this one's chromosome 20. They're flip-flopping entire sections, so now I have genes moved around within the DNA, and it might impact how it's read and how it's used. So sometimes these aren't so good. But ultimately, how many nucleotides are in human DNA, just to kind of give you a little background on these mutations and if you should be worried about them or not. We have about 3.2 billion nucleotides in our DNA, and we code for somewhere between 30 and 40,000 genes. So we can make somewhere, realistically speaking, somewhere between 30 and 40,000 proteins. Now, with that being said, most of our DNA does not code for a protein. It's called filler DNA for the purposes of this class. It does have some functions, but most of it is really not a protein-based function, so it really doesn't totally impact the organism in a long-term fashion. So 99% of your DNA is kind of just there. Some of it is junk DNA that's been held over millions and millions and millions of years from being replicated from different sources and lineages and families, and some of it is kind of just random chunks that are just in there and duplicates and all kinds of stuff. So a lot of it doesn't get reused. So if you have a mutation that occurred at any of those points, really it doesn't matter because it doesn't code for a protein anyway. So any small changes in that DNA really doesn't have broad implications. That's a quick rundown of Unit 5. I apologize for kind of tripping over my words at different points. I hope it didn't limit your ability to understand this. Make sure you reach out to me if you have any questions. That's all I have for today, and have a good one.